This is podcast number 35. Okay, so up until this point in the class, we've been talking about cells, mostly eukaryotic cells, and we've been talking about generic cells, mostly animal cells, some plant cells, but just sort of a general ovoid sort of shaped cell. For this last bit of the class, we're going to talk about specialized cells. These are eukaryotic cells just like any other cells, except they have some specialized morphology, specialized shape, and with that comes some specialized function. And there are great numbers of biologists studying a specific cell type. So for these two podcasts, we're going to talk about neurons. I highly encourage you to take a class from Dr. Renna, who's a neurobiologist, and he specializes in these types of cells. Neurobiologists are rapidly expanding their ranks in biology. When we used to have in-person conferences, the Society for Neurobiology would hold their conferences in stadiums, and there would be like 30,000 people there. So it's a really rapidly advancing field. Okay. A general neuron would be a cell body that has the parts that we've been talking about: a nucleus, Golgi, mitochondria, ribosomes, etc. And then extensions of the cell bodies on one side called dendrites, and then usually a large extension or many large extensions coming from the cell body and away from it called an axon that would be connecting to another cell through a synapse, either chemically or electrically. Chemically more common, where this cell in this diagram is releasing a chemical, a neurotransmitter that travels across a small distance between cells and then is picked up by a receptor on a receiving cell, a postsynaptic cell, causing something to happen. And these are so-called excitable cells. They transmit signals that are important for movement, for secretion of hormones, for development, for all sorts of life processes. A lot of the basics of how neurons work was figured out in this model organism, the squid. So model organism is a model for other organisms in that it's thought to be representative of how organisms work in general, and yet there's something about this organism, or several things about this organism, that makes it easier to study. So in the case of squid, their cells make axons that are really big. And so in the early days of studying axons, we had less precise equipment, and so a lot easier to study axons that are big, and so-called squid giant axon. And then those general principles were then transferred to other sorts of cells that we're interested in, such as mammalian axons. Just some anatomy stuff. When someone says a nerve, like your sciatic nerve or you're getting on my nerves, what they're talking about is a bundle of axons. These axons can be in bundles that are covered with connective tissue, and then supplied by blood vessels. So a neuron and a nerve and an axon aren't the same thing. A nerve is a bundle of axons. Okay. How these neurons are studied, and this was true in the squid giant axon, is through basic electrical equipment that is not that different than a multimeter that you may have in at your house, in a shop or something like that, where there are electrodes that are measuring the potential difference between two surfaces or two solutions, and then that is used to detect signaling. It's a lot more complicated than that, and if you ever get a chance to visit Dr. Renner's lab, he's got a lot of really cool equipment. But essentially, this is the idea, in that you have two electrodes, one measuring a change in voltage or a change in current. Um, Between those two, the signals are very small, and so they go through an amplifier, and then you have to have some way of visualizing what is happening with those signals. So in the old days, it was a chart that was drawing a graph on a piece of paper, and of course these days it's on a computer. And in a cell, you can have your reference electrode outside the cell and your microelectrode inside the cell, and you're measuring the potential difference across those. And the cell is contiguous with the axon. The axon is a process of the cell, so you can measure what's happening in axons by doing this. And so before these microelectrodes became, before we had the technology to make them very, very small, and now we can make them so small that they're the size of just one protein channel. But before that, having an axon that was really big was a big help. Okay. A common sort of bread and butter experiment that neurobiologists do, and Dr. Renner's lab does this, is, in fact, if you get a chance to talk to Matt, one of his students, he's doing this routinely. It's something called a patch clamp, where instead of trying to record from a whole cell, in fact, you're trying to record from just a single channel in the cell membrane that is in a patch of the cell membrane. So these micropipettes are made of glass, and they can be precisely drawn down by heating up the glass, so that the end of this pipette is very, very small, on the order of the size of not much bigger than a single protein that's embedded in the membrane. And then they're placed against the surface of the membrane. The membrane is charged because of those phospholipid head groups, and the micropipette is also charged, and so they form a very good seal, electrical seal. It's called a gigaohm seal, so that there aren't going to be ions leaking around this electrode, so that if you measure a change in current inside this electrode, that is represented by the current that is going through that channel. So it's not current that is going around the electrode, it's actually current that's in that channel. And in addition, because this is a gigaohm seal, you can change the voltage at that electrode by just saying, dialing it in on your amplifier. So you can say, okay, I've got a reference electrode and my measuring electrode, and then because I've got those two electrodes, I can change the membrane voltage at will, and then I can see how does that affect the behavior of this channel. So here's an example of one of those experiments where the voltage is step changed approximately 40, 50 millivolts over this time period, and then current is measured while that is going on, and you see a deflection and the direction of the deflection is dependent on how you do the patch. So this is current going into the cell or current going out of the cell. So this can be going down or going up. That doesn't matter so much. But what does matter is that it's either on or off, usually. That there's no sort of halfway on or halfway off. So you get this sort of burst of current, and then the channel is off, and you go back to background levels of current. Okay. So this is what it would look like under the microscope. This is in the cell body of a neuron, where this pipette is drawn down and is just touching the surface of the cell membrane and forming that gigaohm seal so that you can get a patch of membrane and then see what is in your membrane. There's all kinds of different sorts of ways this experiment can be manipulated. So this is the sort of thing I described, but you can also apply suction through this micropipette and rip off a piece of membrane if you want to study just that piece in another context. You can gently remove it, and so you're no longer in contact with the cell, but instead you can now change the solution on the outside of this channel and on the inside of this channel versus letting the cell do it. And then you can remove your patch, get it stuck to your pipetter, and then apply a little pressure so that you're sort of flipping the channel around. If you notice the orientation of this channel here, now it's facing the other way. And then you can apply whatever solution on the inside or the outside of your channel. So why would you want to do that in different situations? You don't know necessarily what you've got when you do the patch, because these channels don't come with labels, but you can apply certain drugs to the patch that are specific for, say, potassium channels or sodium channels or calcium channels, and by the behavior of that channel in the patch you can figure out what's happening. So for example, let's say you got a patch and it contains more than you want. It contains maybe calcium and potassium channels. Then you put it in poison to poison all the calcium channels so that you're only looking at potassium channels, something like that. But those are just ideas to give you ideas of how versatile this patch clamping technique is. Okay. It's a H5P question for you. Okay. So we're going to talk about how the cell in a neuron can locally change its ion conductance across a small bit of the membrane in what's called an action potential. And in order to understand that, you need to understand, or just remember, if you've had this in another class, that cells routinely maintain ion gradients. If you look at the concentration of ions inside a cell versus outside the cell, not the same. Sodium is high outside, potassium is high inside, calcium is high outside, low inside, etc. So it's not that those ions are just in equilibrium across the cell membrane. The cell is actively maintaining gradients, and it actively maintains gradients through a combination of the action of pumps, like before we talked about the sodium-potassium pump, and channels that are specific for these specific ions. So like I just said, sodium, here's for squid and here's for mammal, much higher on the outside of the cell than on the inside of the cell. Again, higher on the outside than inside in mammals. Potassium, much higher on the inside versus the outside. higher on the inside versus the outside. Chloride, much higher on the outside than the inside. Calcium is another one that's not on this slide, but it's much higher outside than inside. So how I remember this is usually we're talking about potassium and sodium, and I remember the word kin. So potassium is the symbol K, and then potassium is high inside, and then sodium is the other one. So sodium is high outside. How these ion channels are selective has to do with their structure, and also how they open and close has to do with their structure too. So in their three-dimensional structure, which is shown in this outline of purple, and then their backbone structure in this ribbon inside of that, there are a string of arginine residues called the arginine paddle. And arginine is a positively charged amino acid. Arginine, as a positively charged amino acid, reacts to other charges. So at rest, when the cell is at rest, the cell maintains a negative membrane potential, meaning that the inside of the cell, the balance of all the ions on the inside of the cell is negative. If you add up the charge of all those ions, the inside of the cell is negative relative to the outside. Depends on the book you read and the cell type and all that sort of stuff, but you'll typically get a value of minus 60 to minus 70 if you measure the membrane potential. So what is the inside versus the outside, and the voltage difference between the inside versus the outside, and you'll come up with the inside sum of charges is about 60 millivolts less than the outside. So if the inside is negative relative to the outside and the arginine is positively charged, this part of the protein is attracted to the inside of the cell, which is the cytoplasm, or on this diagram, and most diagrams that we're talking about, that would be on the bottom part of this diagram. And it's not attracted to the top part of the cell. Okay? During an action potential, which we'll talk about shortly, the inside is going to become positive for a short amount of time, and the outside negative for a short amount of time. So at rest, the inside is negative, and these positively charged are attracted to the inside. But during an action potential, that flips, and these positive charges are going to shift up and be attracted to the extracellular surface at some point for a short amount of time. And because they're connected to the rest of the protein changes shape. And so you go from a closed structure at rest that looks like this to an open structure because of this shift in the arginine paddle, and that shift is caused by a change in the membrane voltage, and the arginine's reacting to that change. So it's sort of like a magnet. If you shift the magnetic force, this thing is going to be attracted to this side or that side. And because it's connected to the rest of the protein, it changes the shape of the protein. So it's quite literally a molecular switch. The other part of these channels is that they are selective for given ions. So a potassium ion doesn't flow through a sodium ion channel, and vice versa. And that's because the potassium ion is going to be surrounded by water molecules and will be a certain size when it's surrounded by water molecules. That's its hydrated radius. And at that size, those water molecules are going to interact with the lining of a channel, a potassium channel. The amino acids in the lining of that channel are spaced just right for potassium plus its hydrated radius, its water molecules. and another ion, such as magnesium or calcium, are not the right size. And that is how a channel can be specific for a given ion. And that you can have potassium channels or sodium channels or calcium channels, and they only conduct those ions. Okay. Again, just showing you the difference between closed and open. And it's not, it's, if you look at the overall structure of the protein, it has changed, and this thing does look different than that. But if you look at just the channel itself, it's not like it's wide open at this point. It's slightly open, and then these golden amino acid residues here are the ones that are going to interact with the water molecules of the hydrated ion as the channel is open. Okay, so here's an action potential. You've seen this diagram in other classes probably multiple times. So what is happening is at rest, when there is no action potential occurring, the cell is 60 millivolts negative on the outside relative to or negative on the inside relative to the outside, and that's called its resting potential. And again, if you measure a bunch of different cells, they're not going to all be at minus 60. And if you look in a bunch of different books, they might not all talk about being at minus 60. But for a generic neuron, minus 60 is considered the resting membrane potential. Okay? And then what happens is something will occur that causes a sodium channel to open. So what is that? Well, it could be that another action potential has arrived on this cell. It could be a mechanical stimulus. It could be a hormone. It could be a change in gene expression. It could be a chemical neurotransmitter that is causing these sodium channels to open. Some sort of stimulus causes a sodium channel to be open. Normally, if we were face to face, I would ask you to close your eyes and rub on your eyeballs with your fingers, and you will see flashes of light, even though your eyes are closed. So you're mechanically stimulating the neurons that sense light in your retina, and your brain is interpreting that as light, even though they're being mechanically stimulated. All right, you can open your eyes again. So something opens the sodium channel, and that causes other sodium channels to open. And there's a flood of sodium into the cell. Okay? So I should say now that it's not that the sodium is filling up the whole cell. This is a very local change at a little spot on the membrane. And if you measured the whole concentration of sodium inside and outside the cell during an action potential, you would have a very hard time measuring a difference. But at this very local spot on the membrane, you can measure an influx of sodium. And because sodium is positively charged and sodium is rushing into the cell, because sodium is high outside, low inside, it's prevented from getting in the cell because normally this is closed. Something opened the sodium channel, and now sodium is flooding in. Positively charged ions are flooding in, and so the local membrane potential at that spot goes up and goes from negative to positive. Okay? At some point, there is, and this is called a voltage-gated channel. So something changed the membrane voltage of that channel. Again, lots of different stimulants. And the sodium channel is reacting to that change in voltage by opening. Its gate is voltage gated. So there are also ligand gated channels where something binds to the channel, a ligand, and that's what opens it. In this case, it's a voltage gated channel. The potassium channel is also voltage gated, and it reacts to this positive change in local membrane voltage, and it opens when it gets to a certain amount of positivity. Then potassium channels open. Potassium is high inside the cell and low outside the cell. So now you have potassium, which is also positively charged, rushing out of the cell. And that causes the membrane voltage to go down locally at that spot. So at rest, it's negative. A bunch of sodium rushes in at that spot, becomes positive. A bunch of potassium rushes out at that spot. It starts to become negative again. This is called repolarizing. So at this point, it's polarized. This is depolarizing, and then this is repolarizing. You notice that it overshoots a little bit. So it doesn't go back to rest, but it actually goes a little bit below rest, in that it becomes hyperpolarized past resting membrane voltage into the minus 80 plus range. Why does it do that? It just does. That's just the behavior of the potassium channels. And then they will shut because they are voltage gated. They will sense that there's been another change in voltage, and they are shut at rest, and you've gone past rest. And then you will activate your sodium and potassium ATPase, and you'll balance out those ions so that you're back at resting potential. This whole thing, okay, sodium rushes in, potassium rushes out, get back to rest. This whole thing happens over about 4 to 5 milliseconds, 4 to 5 1,000ths of a second. So it's very fast. So first you have the sodium coming into the cell, and then you have the potassium leaving the cell. The sodium coming into the cell happens over a shorter time span, 3 milliseconds, and the potassium leaving the cell, most of that is done by 4 milliseconds, but then a couple more milliseconds are used to pump out the extra potassium and return to resting membrane potential. Okay, so this will start to happen, let's say, at the dendrites, okay, where you have sodium channels open and you start to get this signal. Then that incoming sodium that is coming in at the dendrites will tend to change the conductance of sodium right next to it, because those sodium channels are also voltage dependent, and you're changing this local voltage of the membrane by those sodium channels opening. That causes sodium channels right next to them to open because they're sensitive to that voltage change, and that causes sodium channels right next to them to open, and that causes a voltage change. And then there's a passive spread across the cell body until it reaches the axon hillock, this sort of neck of the axon. And then you'll see action potentials at discrete spots down the neuron, if this is a myelinated neuron. So I said a bunch of words there that I need to explain. If we look at an unmyelinated neuron, so this is just explaining how passive depolarization can spread. Sodium is coming into this local spot on the membrane. That means positive charges are rushing in. That affects sodium channels that are right next to it because they are responsive to voltage, and I just changed the voltage right here. So then right next to it, that's going to respond to that voltage, and more sodium is going to come in. And then that's going to respond to that voltage, and more sodium is going to come in. And that's going to respond to that voltage, and more sodium is going to come in. By the time the sodium channels down here are opening, these potassium channels are starting to open to repolarize this part of the cell, which got depolarized up here. Okay, so this is passive spreading of the signal down the membrane. Okay. There's also another phenomenon that is happening called the refractory period. So after a channel opens, there's another domain of the channel that contains this ball and chain sort of configuration. It actually is called ball and chain. And the after the opening period, the ball will plug the channel on its chain and prevent it from being opened again for a short amount of time while that ball peptide is in there. So that's called the refractory period. After a sodium channel or a potassium channel is opened, there is a period of time where it can't be opened again while that ball peptide is in there. This is really important for the direction of the channel opening. So in this scenario, you see that the direction of the signal is moving in this direction. And because these sodium channels, after they open, get inactivated, even though the charges, the positive charges of sodium that are coming in could go in both directions by diffusion, if they go backwards, they can be there, but because those sodium channels are inactivated, no more sodium is going to come in at that point. And if they're also going to spread in this direction, and these sodium channels haven't been recently activated, so they can open. This refractory period means that the signal only goes in one direction, and that's away from the cell body. Okay. This is another diagram that talks about this refractory period, and now this is adding a myelin sheath, and I'll talk about that in a second. So sodium channels allow sodium to enter the cell. That sodium will diffuse in both directions, okay? As it diffuses in this direction, these sodium channels haven't been opened before recently, and so they can open. And then it'll diffuse in both directions. See the gradient of red here. And then these sodium channels here can open, but these cannot because they are in the refractory period. They just opened. So if sodium floods back and diffuses back to affect these sodium channels, doesn't matter. They're in the refractory period. They're not going to open. Same is true for these guys back there. And so the only possibility is to open up sodium channels further on down the line. Okay? Now, the myelin sheath, let's talk about that. So there is a separate cell that is different from the neuron called a Schwann cell. And these Schwann cells are essentially lots of membrane and not much else. They have organelles and all that sort of thing, but they really specialize in membrane. And that membrane wraps around a neuron axon over and over and over again, meaning many, 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 many layers of membrane. And that membrane has lipid in it, and lipid is an excellent insulator. So that where axons are covered up with myelin from these Schwann cells, the ions can't leak out at that spot. They can only move in and out of the axon at places where one Schwann cell meets up with another Schwann cell. That's called the node of Ranvier. So there's a little bit of naked membrane in the axon at that node, and then the rest is covered up with this Schwann cell. So ion movement happens at those nodes, and it doesn't move across the membranes where it's covered up with a Schwann cell, because the Schwann cell is a really good insulator, prevents that ion movement from happening. So in this diagram, they're showing you the purple. That's the part of the membrane that's covered up with Schwann cells in this material called myelin. So ions aren't going to move in and out at that point. They can only move in and out where one group of Schwann cells is meeting up with another group of Schwann cells at this node of Ranvier. So sodium can come in at this part. It can diffuse in both directions, but it really is only has an opportunity to exchange with the outside of the cell at the node, because at the node it's uncovered, and between the nodes it is covered. Just show you this picture. This is a cross section to give you an idea. Here's the axon, and here's the layers of myelin, and here's the Schwann cell that has laid down this multiple areas of membrane. So you can see how this would be very well insulated, and it would be hard for ions to get in and out over that part. Okay. So this is just another diagram to show you that there are multiple connections made with the cell through these myelin layers. The sodium channels are in the node. The potassium channels are near the node, okay, but not actually right at the node, and still has the opportunity to exchange sodium and potassium at this at the node. But you don't see sodium channels back here, or potassium channels back here in the middle of something that would be covered with myelin. But there is more coverage on the potassium side than on the sodium side in terms of connection with the outside of the cell. Okay, so Dr. Renna has a very useful analogy. This is also detailed in your book. If you're thinking about delivering water at the end of a garden hose, and the rate at which that water is delivered at the end of that garden hose, let's say you are filling up a hot tub or something like that, and you need to have water delivered at the end of that hot tub. If you have a garden hose that is full of holes, like you can see here, that's going to affect how much water comes out the other end, because you're going to lose a bunch of water along the way. Okay, and so the speed at which the signal is arriving at the other end is slowed down just because you're losing a lot of the signal along the way. Okay, that would be an unmyelinated axon. Okay, now myelin would be as if you are covering up most of these holes, not all of them, because you still have to have some exchange at the nodes. But most of these holes in the hose are covered up by duct tape. And when you do that, now your flow at the end of that garden hose improves dramatically. You get a lot more signal out the other end of that. And so if you're measuring the rate at which a signal goes from one end of the axon to the other, or one end of the hose to the other, if you're covered up with myelin, you go much faster than if you're not covered up with myelin.